The University Science Headstart lecture. My name is Brianna Ragel, or Bri. You might have come across me before if you've attended particular ones of the VCE science lectures. In terms of about me being quite relevant to this lecture, I graduated from a Bachelor of Science Advanced Global Challenges Honours at Monash. Basically, it was kind of like a normal Bachelor of Science, except 1/4 of it was focused on impact through science, and some other differences along the way. But most of my classes were with all of the standard science students. With that, I did also at one point become the president of the Monash Advanced Science and Science Scholars Society. So if you have questions about clubs and societies or social life at uni, what that can look like, I'm definitely happy to answer those as well. I majored in ecology and conservation biology with a minor in mathematical statistics. So my first year looked like doing advanced chemistry, biology, same biology that the more human biology focused people were doing, and also a bit of maths. So depending on what you're doing, you might have slightly different units that you're starting off the year with, or depending on what university you're at, they might give you a different name like subjects or courses, but the fundamental content that you tend to cover in first year science are focused on the biology, chemistry side of things for our first block, being that those are very popular subjects to start off with. And then the second half that we're looking at goes into data and some general guidance about science. That's going to be very applicable to everyone, regardless of what courses you've picked. Now, with our uni notes free resources that we have, since we're starting off the uni notes and looking at these lectures that obviously you know about, we also have other things that you can get access to as well, which I'll talk a bit about throughout the lecture. So we've got study notes that you can download for free based on the things that people are struggling with in these lectures. I'll add some extra ones into there as well. There's the community where you can have discussion, help to talk to other people, videos, news, all sorts of things. And subject reviews is something that you might not be familiar with if you're coming at this from a high school perspective. So that's something that's actually been part of the ATAR notes, or previously the forums, and now we're onto the new website. But it's been there for a while, where you'd have uni students coming giving feedback on their course and that kind of specific advice to people who are going to take it. Now, as always, we tend to have this practice of wanting engagement and for us to be talking to each other. Since we've got these new resources coming out, I'm going to start off by asking what resource you would be most interested in. Okay, great, we're starting to get some answers through. Right, so we've definitely got divided opinion at the moment. Green. So I'll talk a little bit to you about what these look like as it goes through. So I'm seeing a fair bit of interest in free notes. Please do feel free to comment in when we have that space after this poll, and let me know any particular parts of the lecture that you struggle with, which I'll try and pick up from the questions as well, so we can be more targeted towards that. I can see these other aspects there. Okay, so I'll leave this a little longer, but I'll swap back to our slides to go through this a bit more. In terms of the uni guidebooks that you can see, this is a kind of preview of what they can look like. So you've got content summaries for core subjects written by recent university graduates from across Australia, covering the fundamentals for a range of degrees. Again, in something like science, which is a quite generalist degree, you're going to have a range of different specifics that people go in, but really focusing on those fundamentals. If you want to keep updated with this stuff, really advise you to go over to uninotes.com and keep on that. Alright, let's close this off because I think we've gotten the kind of answers that we're going to get. And head into our lecture. So as per usual, we've got the slides. I'm going to try to make this really engaging, lots of questions. Really encourage you to ask any questions you have. Again, even if it's about things that seem silly, like, what your day kind of looks like, how you handle the travel, whatever it might be, this is your chance to kind of get those questions answered by someone who's, gone through the full undergrad plus honours process on there. Or if you're like, I don't understand what you're saying with undergrad or honours, that kind of thing, seriously, no silly questions. Just whatever you have. In terms of the specifics of what we're going through topic-wise, I mentioned that biology and chemistry being really popular topics and that I'm focusing in on those for our first part. In terms of being a bit more particular, the chemistry we're going to be looking at is more on that quantum chemistry lens. So we've got a bit of overlap with physics there. This is something that when I was studying was the kind of first things covered, and I know from people who have recently gone through that, it's still something that you have at the start where you're looking at that, particularly for chemistry students without a physics background. That can be, a bit difficult. And then with biology, we're focusing on taxonomy. So whatever path you want to go in biology, we're going to be looking at how we go from the idea of life in general down to what makes a mammal, right? In your kind of high school studies of biology, if you've done that, we tend to look more at that bit of how are humans different from other primates? Here we're going to be looking at that bigger picture overall. From our break that we'll have in the middle, again then data and stats, hugely important regardless of what major or minor you might want to have, and then some general guidance. So let's take a look at chemistry and this idea of quantum chemistry. It's not really going to be like when you're going, movie and they just throw around the word quantum all the time, but looking at this idea more, digging into electron configuration that you may have covered, particularly in year 11. So we're going to start off with this idea of orbitals. So we've got these different energy levels that electrons can have around an atom. So we've got these major energy levels, which we call shells. And then we also have these subshells. So we've got S, P, D, and F, each of which can only hold a certain number of orbitals. Then each orbital, which is at a particular energy level, can hold 2 electrons. And so it might only have 1 in it because, it doesn't need to be holding 2 at any time, but a maximum of 2 electrons in any given orbital. Total number of electrons you have per shell is 2n squared. So we know that our first shell is 2, and then we go on to 8, and we continue getting larger. What we're going to be looking at, or at least part of what we're looking at in this idea of subshells, is why it is that our S subshell only holds 1 orbital, why it is that our P subshell has 3 orbitals. We're not going to focus too much on D and F, but we're going to be digging a bit more into the why. So just in case I was a bit unclear for anyone, when we're looking at these orbitals, that still works within that kind of shell system, but in terms of the graphical representation, it's a bit different. So you can see the S orbital has that spherical shape, and that P orbital is that more polar orientation. So we're needing to look at that 3-dimensional space. Here it's shown broken apart, but in reality, we have this shaded region where we can, expect the electrons to be 95% of the time. That's all going to be centered around where the nucleus is. You do want to make sure that you know how to write electron configuration. So again, for some of you, this may be thinking back to year 11 or to previous studies where you're going, all right, 1s2, 2s2, 2p6, 3s2, and needing to continue along in that way. they absolutely can, and very likely will, ask you to write the electron configuration of a given atom or ion. A trick you can use when writing condensed electron configurations is to take one of your noble gases, and then you write that in the square brackets. And so when you do that, you're saying, okay, well, the electron configuration of this, and then you just add on the extra bits belonging to your particular atom. So when we're writing it in the expanded form, it's going to look something like this. But if we wanted to, we could condense that by using our different noble gases to write the condensed electron formula. So if you just Google the electron configuration of something, it probably will give you that in the condensed form. Rather than just writing them out on a page, we also want to start to get the idea of electron configuration in terms of the energy levels, and so we're looking at drawing out these orbital diagrams. So generally you wouldn't be expected to do one this big, and often they might have 2p in the middle and then kind of show them on either side rather than branching out to the right. But you really want to note that down the bottom is where we have the lowest energy, and then you work your way up. So we have different principles that we need to be following when we're filling out our diagrams. And you will be expected to know, the name of these and what they mean, as well as being able to actually apply them in the diagram. So we've got the off-bow principle, which is where electrons are filled into atomic orbitals from lowest energy to highest. So really, you're working your way up the page. Then we've got Hund's rule, where every orbital in a subshell is singularly occupied before any orbital is doubly occupied. So when you're writing it out, if you look at that 2p level, for example, you would need to put 1 in each of those 3 before you double up in any of them. Then you've got polarized exclusion principle, which says that no two electrons can have the same quantum number. We're going to go into the different quantum numbers and what that means when we're talking about that. But essentially what that's going to mean is you can't put 3 electrons on one line. You can only have 2 electrons in any orbital, and they have to have opposite spin. So you'll do one as an up arrow and one as a down arrow. So when we look at these quantum numbers, we have a few different ones to be aware of. There's the principal quantum number, n, which is describing the size of the orbital. So this is all about what you would kind of normally think when it comes to how our electrons are arranged in their different shells. For this, you're going to have positive integers only, so like 1, 2, 3, 4, 5, 6, continuing up. Then we're introducing a couple of new concepts. So we have the angular quantum number L, which describes the shape of the orbital. Essentially, this is telling you, is it an S, is it P, is it D, is it F? So for this, the integers are between 0 and n minus 1. So you'll note that when we look at our first shell, our first electron shell can only hold 2 atoms. It only has that s orbital, and we're told that again here in terms of there's only one angular quantum number. So it looks like there's a typo in there with the 0 for spherical. Then we've got the magnetic quantum number, m, which describes the orientation of the orbital. So these are integers between negative L to positive L. So depending on what type of orbital shape we have, you have a wide range in how many different options you have here. Then finally we are looking at the spin. So arbitrarily this is kind of set to 1, there being plus half and minus half. The really key thing is that it's 2 different options, which is why we can have 2 electrons in the one orbital, because even if everything else is the same, we can differentiate them by their spin. So my question here is, how can we explain that the s orbital, we can only hold one orbital, so we can have a maximum of 2 electrons in there, whereas something like E can have 6 electrons due to having 3. Don't worry about getting it wrong, just take a guess, and if the answers coming up are really like, showing that you're not sure of what the answer is, I'll go through and I'll explain this again a bit more slowly, go over it a bit more, because it is new concepts. Right, great, we've got our first answer in. Right, so if we think about this, the s orbital, if we go back to that picture of it before, it's a sphere. Right, so we can't really orient it in a different way. And that's reflected by the fact that we only have one number that we get out when we look at the number of possible orientations. Because all quantum numbers need to be unique, we can't just double up and add an extra orbital in that has all the same properties. We would need there to be something different. And so we can fit 2 electrons in because the 2 electrons can have 2 different possible spin values, which means that they can still be unique because we'll just give them a different spin even though everything else is the same. So the really important ones here are the all quantum numbers needing to be unique, and which then plays in with the fact that we get 2 electrons because there's 2 possible spin values, and that we only have one possible orientation, and we only have one number when it comes to what the s orbital can be for a given principal number, right? For a given shell that it's in. So all of the options attribute to this. This is the best answer on here. So when we fill out an orbital diagram, usually it's not going to be in that circle, it'll be more the straight line, but we have this up arrow, down arrow to indicate the different spins. You want to fill in one subshell at a time, starting with 1 electron in each orbital with the same spin. Right, and then work your way from low energy to high energy until the amount of electrons is what's required for your atom or ion. So if we look at this and I was filling it out, I would fill in 1s first, up arrow, down arrow, then 2s, up arrow, down arrow. Then when I go into 2p, I would go for each orbital, like up arrow, up arrow, up arrow. And then I would fill in the down arrow, down arrow, down arrow. Right, so just be careful with the order in which you fill this out. Make sure that you're very careful to be the same as examples you're given. I would recommend really not changing things, because even if it seems like, it's a bit arbitrary, you'd really want to be quite confident before you make something different from like a sample diagram. And then my question that I have here is, without being told or looking at, the link from the file, how could you figure out what atom this orbital diagram's for? Right, so imagine you've got access to the periodic table. Right, so we're seeing some answers coming in here. So yes, we absolutely can count the number of electrons. We just count our arrows, check the periodic table for which atomic number it matches. Could throw us off a little bit if it's an ion, which is why technically I put guess in there instead of being like no for sure. But absolutely that's the approach that I would take. There's also another thing that you could look at. Yep, count the electrons, match atomic number, absolutely. There is a kind of fast way. You don't necessarily have to count all of the electrons. Right, so this is starting to get a bit close where we're looking at the fact that we've got this 4 here. So I'm assuming SF, just a bit of a typo. If we look at the fact that its last one there is that S, and it's got 1 electron in that, that's telling us it's a group 1 element, and then we look at that 4, and we can see the period. So we could also identify it quickly that way, instead of counting up each individual electron. But either technique will work. An important thing is that we do have exceptions to our usual electron configuration. If you think back to that kind of original orbital diagram, sometimes the lines look pretty close to each other. And so that means that they have a similar energy level, and we have a more stable configuration in the D shell if it's either half full or completely full. So that stability is associated with a lower energy level, which then means that we're going to be filling up our orbital in a slightly different order. So sometimes an electron will move from one shell to another, from 4s to 3d, in order to make that D shell more stable, because there's a very small energy difference between the 3d and the 4s orbitals. So chromium, you might think that it's going to have, so here it goes up 1s2, 2s2, 2p6, 3s2, 3p6 like we'd expect. Instead of going 3d4, 4s2, we've got 3d5, 4s1. Right, so as we're filling it up, we want that 3d5 so that it's half full, even though that means that we have to tweak things a little bit. Same thing happens with copper essentially, except this time instead of it being about it being half full, it's about it being completely full. So our correct configuration for copper, we have 3d10, 4s1. So let's have a look at what these orbitals might look like. Imagine you've got your nucleus in the center of those axes. So if we're drawing out our p orbitals, it will look like this. We've got our 3 different orientations. We can also refer back to the quantum number of 1, and we know the orientations go from negative L to L, so that's negative 1, 0, 1, which gives us those 3 orientations. So again, we can link back those quantum numbers to the results that we actually see to explain why we have 6 electrons in the p orbitals. Now the neat property when we're looking at this is to then apply it across to our visible colours. So when our atoms are heated, they can produce light, which is made into an emission spectra. And we've got our unique emission spectra. We've got electrons absorbing energy, can jump to a higher energy level, the excited state. So if you look at the electron configuration and it seems incorrect, it's not following the Aufbau principle, we've got things at a higher energy level before filling up the lower energy levels, that might be because it's been excited. This is unstable, so the electrons are going to return to the ground state where they're at those low energy levels. In this process, when we've got that energy release, we're releasing energy in the form of a photon. So we've got our photon emission, and then the energy of the photon we can use to find the frequency of the photon. So the energy gap between the orbitals tells us the energy of the photon that's released. So that's what's responsible for our different colours we get in flame tests. And we can see this example with sodium here, where we have our 3s electron jumping up to 3p. And there's going to be multiple different possible excited state configurations depending on how much energy is absorbed. We can use this to predict what colour a metal is going to produce when we heat it. So we know that the emitted photon will have some energy level. That energy level will be Planck's constant, which is just a number, multiplied by the frequency. And the frequency of light tells us the colour. So when we're looking at this, a couple of things to be wary of. Planck's constant comes in two different versions. We've got the electron volts version and the joules version. These are different numbers depending on what units you're using. Make sure you pick the one that matches up with the energy you're given. So if you're given energy in joules, use the version of Planck's constant. This is the one starting with 6, that's in joules. If you're given it in electron volts, then you want to make sure you use the electron volts version of Planck's constant. So a question on that might look something like this, and then if it was something with a visible colour, it might present you with a colour wheel. So here we're told a hydrogen atom has an electron transition from an orbital with energy 12.8 electron volts to 10.2 electron volts. We're just going to take our 12.8 minus 10.2, and that tells us that the energy difference is 2.6. Now we're told that Planck's constant is 4.14 times 10 to the negative 15 electron volt seconds. Okay, so we're using the correct version here, and we need to know the frequency of the emitted atom. So E equals HF, quick rearrangement, E, and then we're just going to divide that. Again, as per usual, just to make sure you're careful with brackets on your calculator. And then you'll get out, in this case, that we have 6.28 times 10 to the 14 hertz. So that was our kind of calculation example. Now I have a question for you. In a flame test, copper is observed to produce a blue flame, whereas lithium produces a red flame. Which metal emitted higher energy photons? Great, first answer in. Alright, I'll give you a quick look at what the answers are looking like before we go back to seeing this. Okay, so I'll start to talk through the steps of this. We're comparing a blue flame versus a red flame. So we look at our visible spectrum and we can see that blue is higher frequency and lower wavelength compared to red. We know from our equation before, E equals HF, that higher frequency is associated with more energy. And so we're going to use that to then go, well, since the blue is a higher frequency colour, it's got more energy, and therefore it's the copper that has produced the higher energy photons, as has been indicated here. Right, so good job for everyone who answered there. I'm going to briefly, and this is kind of coming at the end of this quantum chemistry topic, talk about the fact that this has implications not just for flame tests, but also for other things. So we can look at these coordination compounds. We've got a coordination complex where we have some like metal in the middle surrounded by other things. So in this example, we have water molecules that are surrounding this metal. And so that negative oxygen is being attracted towards that positively charged metal ion. And then we've got this bonding interplay that occurs there. And so the way that the ligands, and particularly the ligand donor atom, so in the water case the oxygen, interact with the metal center is going to impact the orbitals that we have on the metals. So if we replace the ligands with something else, we can change those energy levels, and thus we can change the color of the compound. So you can just directly see this in solution. If, for example, you change what's in there, change the equilibrium to displace the existing ligands, you can see that color change directly. So cobalt can be a quite good example of that, to see the changes. Again, I'm not really expecting a huge depth on this because we're going quickly onto this, but this is to say that the atomic orbital work that you do, where you look at one element and you look at its orbitals, you then expand on to look at molecular orbitals, and then you can look at these coordination complexes. So it does build up, and you can later look at other things in relation to that as well. Further impacts. Alright, we are now on our biology section. I can't see any questions, so we're going to press on with bio. And particularly we're going to need a strong understanding of phylogeny for this. So generally when you're starting out doing biology in your science course, you're probably not actually going to start with this. You're probably going to start looking at your macromolecules, like how do proteins work, how do carbohydrates work, how does DNA is made of nucleic acids, those kinds of things. I've chosen to focus on this in this lecture because this is something that does actually become important, and it will be useful for whatever biology field people go into in terms of having that background understanding, and it's also something that people tend to struggle with more. So for people who haven't done biology in VCE who then do biology at university, there can be a bit of that struggle in terms of looking at polymers and how they work, but particularly for everyone, regardless of whether they've done bio or not, this often tends to be a topic that trips up people. That and also looking at plant life cycles can be a bit trippy. So we're focusing in on taxonomy. Again, like with other science subjects, remember to break words apart for their meaning. When you come across new terminology, it's not a bad idea to look up where it comes from, so then you can use that to build up that bank. Particularly when you're looking at a field that you want to go more into, the more familiar you can get with the root meanings behind the words and those components, that's going to help you when you come across new terminology. Sometimes you're going to be able to just intuit what it means based on those root meanings. So, as I said before in the intro, we're going to be going from all life down to mammals, describing some of these different key distinctions that we have. But first, let's talk a little bit about our terminology. So we have the idea of being monophyletic. Mono meaning one, you've got the common ancestor and all descendants of the common ancestor in that grouping. Whereas you can look at a paraphyletic group, which contains a common ancestor and some but not all descendants of that common ancestor, or a polyphyletic group, which does not contain a common ancestor of the included taxa. So when we're trying to classify things in biology, sometimes people can be a fan of, in the past we tend to look more at phylogeny, right? So you might go, oh, mammals are hairy and they produce milk. Then you might go, hang on, does that make a coconut a mammal? So what the trend has been more towards is looking at evolutionary relationships and building up these monophyletic groups. So sometimes this will be called cladistics, and we'll talk about having things in a particular clade. So those terms can be used interchangeably when we're looking at a clade and these monophyletic groups. So to start off with, What type of grouping would B and C, without including any of the common ancestors, be? So we're just looking at descendants BC, and none of those ancestors are present in the forks. Right, so remember when we're looking at any of this kind of tree, we've got some descendant here that branches off into different paths, then we've got some ancestor here, until eventually we get the extant species, the ones that exist now. Right, to give you a glimpse here, we're having different answers. I will give you a hint and say that the most popular answer is not the right one. For this question, we're just looking at these two. Again, at the moment, paraphyletic is the most popular answer. It is not correct. Right, this is why I do the practice questions on this, because this trips people up, particularly paraphyletic versus polyphyletic. Right, yeah, we're definitely getting closer here. This time it is safe to follow the crowd because polyphyletic is correct. We're not including any of the ancestors here. If it was paraphyletic, I'm just gonna... If it was monophyletic, it would, and we had B and C in there, it would need to be this whole giant group. Right, forgive the messy ink there. Because if we're including B and C, we also need to include all of their common, most recent common ancestors up until here. If it was paraphyletic, it might be something like, including everything there and all the ancestors, but not A. So to confirm again, the correct answer is polyphyletic. We're going to do another practice question on this. So I'm going to highlight the relevant ones here. So what type of grouping would C, D, E, the most recent common ancestor of C and D, so that's this guy here, and the most recent common ancestor of C, D, and E, so that's this one here. What would we call that grouping? So in other words, we're taking this taxa here and all of the descendants of that. Alright, again, paraphyletic is popular. It is not correct. Alright, so I'm showing this so you can see, not on your own. This is a tricky subject. Paraphyletic is not the correct answer. Paraphyletic might be if, say, I was taking this whole thing except E. But in this case, I'm taking taxa and then all of the descendants together. Exactly, I can see someone switched across to the correct answer. Right, so one thing and all of its descendants, that makes it a monophyletic group. So I'm going to close this one off. We will do more on this and just head back here. So remember, a monophyletic grouping, a common ancestor and all descendants of that. So if I took this and all of the descendants, that I don't have to take everything that I can see. I just have to make sure that whatever I'm taking, it's all of the descendants of that thing. So if I wanted to, I could make another monophyletic group that's just C and D and this ancestor here. That's fine. So I'm choosing to start it from here. As long as I take all of the descendants, it's a monophyletic group. Or for example, I could take A and B and their most recent common ancestor here. And then I'm taking a monophyletic group as well because it's one thing and then all of their descendants. If, on the other hand, I decided to take D, E, this ancestor, and I didn't take C, then this would be a paraphyletic group because I'm taking this round one, and I'm taking most of the descendants, so I'm taking some of them, but I'm not taking all of them. Right? It has to be all of them to be a monophyletic group. Just all of the descendants of a particular point. So here we look at life from the kind of most zoomed out view, where we're looking at our three domains. We've got bacteria, archaea and eukarya. Being eukaryotes, animals are in eukarya, and so we belong to this grouping. So I'm going to check your understanding. Eukarya, is it monophyletic, paraphyletic or polyphyletic from what you see in the diagram? So with the color coding, we can see that we're starting off at here. We've got some ancestor, and then we're taking all of these descendants of that common ancestor. Right, exactly. Alright, we're having a split opinion now. Okay, very undecided. So if I take this and I take all of the descendants, that makes it monophyletic. Right, that's what's happening here. I'm taking everything that's descended from one particular ancestor. I don't have to take the whole tree of life, just as long as whatever ancestor I start at, I take all of the descendants of that. So eukarya is a monophyletic group. If I wanted to make it paraphyletic, what I would do is I would exclude something. Right, so I might say I'm taking all of eukarya except the plants. Now, because I'm not including all of the descendants of that ancestor, now it would be a paraphyletic group. Or if I wanted to make a polyphyletic group, I might do something like taking cyanobacteria and animals and say, okay, well, I'm making a group of them. Right, so for example, cyanobacteria and plants can both photosynthesize. So if I decided to make a grouping based on photosynthetic organisms, that would be polyphyletic. Right, so I can see we've got a question here. Why is it monophyletic if we're not taking all the archaea descendants? We are. So the fact that in the diagram we can see them all in the same colour, we're taking all the animals, all the plants, all the fungi, everything, that makes it monophyletic. If that doesn't fully answer it, please feel free to ask again. When we take this eukaryote group, yeah, every descendant from like this point is being included. Alright, I'm going to add in another question just to check understanding. So the question is, what grouping would ciliates and extreme halophiles be? And again, our options are monophyletic, parophyletic, or polyphyletic. So the ciliates are over here. The extreme halophiles are here, so that something like, you remember from how TAC polymerase comes from Thermus aquaticus in the hot springs? Thermus aquaticus would be an example of our being able to withstand extreme conditions. Alright, exactly, perfect. That would be a polyphyletic grouping. Alright, let's continue. So as we go down and we get a bit more specific, one of the key distinctions we have is whether something is triploblastic. So something that's triploblastic contains 3 germ layers: endoderm, ectoderm, and mesoderm. In humans, the ectoderm develops into skin and nervous system. The mesoderm then develops into a range of things, including muscle, bone, and connective tissue. And the endoderm is that kind of inside layer, right? If you think of humans as being a pretty complicated tube, then the ectoderm is the outside of the tube, the endoderm is the inside lining of the tube, and then the mesoderm is the stuff in the middle. So from early on in our development, we have these different layers, and then they differentiate into all these things, and we get all these complex tissues. So with this, I've kind of skipped into, we're eukaryotes. Let's then zoom into different types of eukaryotes, and let's look at the ones that are triploblastic. Then, of the ones that are triploblastic, we're going to be looking at particular animals where we have bilaterians. So these are all triploblastic because they're all descendants of that group. They're all part of that. And they all have bilateral symmetry. So if we think about the cnidarians that we have just above on this phylogenetic tree, the cnidarians can include things like jellyfish, for example. Alright, so, or some kind of anemone. And if you're thinking about an anemone, it doesn't have that body plan where we've got that mirror image across that bilateral symmetry. So this is an important distinction, and you can see that there's a bunch of things that don't actually fall into this category. Then we're going to zoom in a bit more, and hopefully this makes the triploblast stuff make a bit more sense when we look at the idea of coelomates. So again, these all have to be triploblastic by definition. They're a group within the bilaterians. So we've got some things that are bilaterian, but they're not a coelomate. For example, flatworms have a Bilateral symmetry, they follow that body plan, but they're acoelomates. They don't have a coelom. And if you look at these diagrams, you'll see that the coelom is this kind of gap in the tissue. And so here we've got this pseudocoelom, which isn't contained within the mesoderm, and it's just a gap between the mesoderm and the endoderm. And then we can see for our true coelomates, our eucoelomates, they have this coelom, this cavity within that mesoderm. And so it's going to be fluid filled, which can be great for things like using hydrostatic pressure to move around. Then within our coelomates, we go to the deuterostomes as part of another grouping. So you can see we're looking quite early on in development here. We've got these different layers. So there's the coelom, we've got mesoderm, there's the ectoderm, the outside endoderm. And then we have the formation of these gaps, right? So in a protostome, we have the mouth form first. In a deuterostome, it means mouth second. So the anus is formed first, and then the mouth is formed later. So humans belong in this deuterostome category. And then we get more specific again, saying humans belong to phylum Chordata. So chordates have these 4 key features at some point in their development that define them as being chordates. So there's the notochord, the dorsal hollow nerve cord, pharyngeal slits, and the postanal tail. So if we take a look at what that can look like, you can see those features here. This one might be testing your understanding a bit, but why might humans have pharyngeal slits in embryonic development, despite these not developing into gills like they do in some other chordates, like the bony fishes? Remember, in the chordate definition, you don't have to have them your whole life, but we show that we have this pharyngeal slit evolutionary history through our embryonic development. So what I want you to think about when you're trying to answer this question is our evolutionary history. So for example, right, the postanal tail, you could basically swap that in with the pharyngeal slits for this question, being like, we don't have a tail, but we are chordates, so what's up with that? Right, so we are grown in an embryonic sac that is filled with fluid. That isn't quite the answer that I was looking for here, but it is a kind of valid point to make. In terms of the importance for that for gas exchange, we don't really need the kind of developed pharyngeal slits that go into gills, like in terms of our survival in there, but it's certainly not a bad thought to have. And our kind of next zoom in relates a bit more to the fact that that embryonic sac is important. Alright, I can see someone's typing, so we're just going to give a bit of time here. Again, maybe think of the postanal tail as an example here. We don't have that as a feature that we can observe to adulthood. It is there in our embryonic development, And you'll notice that when you look at pictures of embryos, the kind of earlier you go in development, the more it seems like a whole bunch of other stuff. And as you get later and later, it becomes a bit more human specific, which can include getting rid of things that we don't need anymore. Alright, we'll be trying to answer this and continue on. You can finish up your thought. Alright, I will be kind of checking back and looking at that, and I'll talk while we're a little bit more about these features. So the notochord is that provides structural stability. So not everything here is a vertebrate, but it's important for that development. Yes, there we go. Pharyngeal slits are a sign of our evolutionary history. Perfect. So it's one of those indicators of evolution. I'm going to be a bit nitpicky here because we are still part of phylum Chordata, contained within the Animalia kingdom. So we are still chordates, but it's a sign of our evolution from the earlier chordates. Absolutely. So it's a holdover from our evolutionary history, and it reflects that. So we kind of make these adjustments and get rid of some of the stuff that we have earlier in embryonic development that are residual and we don't really need anymore. So let's look a bit here. We've got our deuterostomes that then branches off into our chordates, which possess a notochord. And then you can see that we've got a whole bunch of different things here and some different categories across on the left. You'll notice that most of these are aquatic. So what might some barriers be when we're looking at that transition from aquatic over to terrestrial? So you'll notice like pretty much everything to like here, and we're looking at it living in the water. And then we've got our amphibians. We get into our amniotes. It doesn't necessarily have to be directly related to this diagram, but if you're an organism that's used to like living in the ocean, your whole evolutionary history is there, what might be a barrier to being on land? Just kind of basic things. Right, limbs instead of fins, right? You want to have a good way to move around. So you can see in some of those ones that are a bit more closely related, we have that move across where we are using more limbs, being able to, or if there are fins, they certainly have more structure behind them. While we wait for that typing to come up, I'm going to go across here to give you some more hints. Think about a difference between potentially amphibians and reptiles and mammals. So that could be one thing to think of. Or if you want, you can go a bit further back and go, why would it be, why would we necessarily care about being vertebrates? What does that mean in terms of land? So you've got a few different options here. Just gonna options. Right, temperature fluctuations in environments, different climate on land as opposed to the ocean. And particularly we're going to be thinking that often on land, you might have things, you've certainly got more extremes, so you might have to worry about things like freezing, which you might be more likely in, going deep water or particularly cold water as used to, but heat is something that's going to be a bit hard, right? And so absolutely you're going to need significant mutations and alterations. A key thing I'll be thinking about with temperature fluctuations, if you're used to an aquatic environment, you think about when we're talking about like fish or these amphibians, right, where they're still laying their eggs in the water, right, because you need to have a way to not have them dry out. And this links to what was being said before about when we think about how a fetus develops in a fluid-filled environment. So a really important adaptation that we have is the amniotic egg. And that's going to mean that we can produce young on land as opposed to in the water. So great answers there. Absolutely, there's a whole bunch of different mutations required. Some of the other ones I was kind of hinting at is that in water, you've got the water pushing up on you, so you might need a bit more structural support if you're wanting to go on land, whether that's from having, vertebrates having that vertebral column, is like endoskeleton way, or you have, like, for example, insects using that exoskeleton. So there's different ways of adapting there, but you will need something. You can see the development of lungs, different air exchange, limbs, as mentioned before, and now the amniotic egg. So with the amniotic egg, you can think of like a chicken egg, for example, right? With this, you've got that hard outer shell which allows air exchange while preventing drying. And so we can have this on land, it's not going to dry out, there's plenty of fluid in there. Now, many mammals do not lay eggs, but we're still part of this monophyletic group. Early mammals did lay eggs. Some mammals, monotremes, still have not lost the ability to lay eggs. And the kind of live birth that we have developed on from the amniotic egg. Alright, we've done a bit of practice on this. We've got another question here. We're now looking at, we've got our tetrapods which have 4 limbs, then we look at our amniotes. Within the amniotes we have reptiles. What kind of grouping is reptiles? And so we can see reptiles, we've got the extant reptiles, so things like currently existing turtles, tortoises, lizards, snakes, crocodiles. We've got ancestors. And yeah, I will tell you that one of the answers at the moment, yep, correct. Alright, most people are on the right track here. We can't call this a monophyletic grouping because if it was monophyletic, it would include the mammals and the birds. Right, particularly the birds are a key one to think about here. If we include some of the mammal ancestors as reptiles, we could more easily decide, you know what, even though they might seem like reptiles, we'll ignore them. But birds we would definitely have to include if we wanted to try and make reptiles a monophyletic group. So with the way reptiles is commonly understood to not include birds, it's paraphyletic. We're taking our ancestor and then we're taking descendants of that, but not all of the descendants, making it a paraphyletic grouping. And now we're on to mammals. So mammals, we've got Mammalia as a class within phylum Chordata. And above that we've got clade amniota. Mammals have mammary glands, so this is where the term mammal has come from, and which can produce milk to feed their young. I've also got some other features in there like cool brain, where we have the neocortex, which is great for our higher order thinking. All of those other things. We are now... So in terms of our topics that we're continuing on to, we have stats and data. Stats is often much maligned by people. They're like, oh no, stats, I don't really understand it. How do I know I'm doing it properly? Like maths, but it is so important. If you are not sure what you want to do and you want to boost your employability, invest in stats and data. If you want to continue on to future research, invest in stats and data. These skills are incredibly important, often underlooked, and I really believe if more people learnt more about this, we would have better science, better scientific community. When you look at the statistics that come out about stats being misused in papers, it's awful. Really, please do pay attention to this. So the further you go into science, the more important stats will become. This is something that's often a bit brushed over when we're looking at the high school level, but as I've said, it's very crucial. You'll need to not only be able to interpret stats such as p-values and standard deviations, but also calculate them using technology. And you might have the same experience that I was having where in chem labs you need to be able to calculate the standard deviation by hand. They give you the equation in the lab manual, but you need to be able to work this stuff out. Make sure you're very careful and precise with your language when you are talking about statistics. You're likely to initially use Excel, right? If you want to calculate the mean, calculate the standard deviation. If you want to pursue research, honestly, even if you don't but you're interested in this stuff, I really recommend learning the basics of a programming language such as R or Python. Python is often touted as a very beginner-friendly programming language. There's a ton of free resources for it. R is a bit more specialized in that it's less in the general world touted as being great for beginners, but R is specifically designed around statistics. Like Python, it's a higher level of abstraction, so it's more closer to regular talking compared to using binary code or that kind of thing. Right, so we're not looking at super intimidating languages here. And if you can learn some of these, but it's so useful, opens up so many doors for you. When I was doing my internship, I had a lot of comments from people about oh, you know some stats and you know how to use R, how beneficial that is for employability as well. So again, something that I really recommend. Now to go into the more standard content, right? So we've got the standard deviation describing the spread of values around the mean. This is something that you've probably come across before, whether in a science subject or in a math subject. And the higher the standard deviation, the greater the variation around the mean. Standard deviation is the square root of the variance and has the same units as the mean. So if we think about standard deviation and the process for calculating this, right, you take your mean and then you subtract your individual data points to get the difference between your mean and that data point. You add all of those up together, square it, divide by the amount of data points. So it really is telling you what the standard deviation is, how much your data points move away from what that mean is. And so that's just a basic, what we call descriptive statistic, which is useful to describe what the data looks like. But often we don't just want to know about the shape of the data, we want to know how we can interpret that data. And so I'm going to put in a concept here called the null hypothesis. So when we're writing a hypothesis, usually people think about the alternative hypothesis, or H1, which is that there is a particular relationship between your variables, right? As angle of launch increases, the projectile will go further, or there'll be greater horizontal displacement, something like that. But the null hypothesis is that there's no relationship between your variables, right? Null. And in scientific investigations, we're interested in whether or not there's sufficient evidence to reject the null hypothesis. So you might have your data which seems to show a difference between these groups, but can you actually say that it's just luck, or maybe there isn't actually a relationship and you just happen to pick ones when they got a higher value in this group compared to the other one? So we'll go on to the different errors we can make in terms of hypothesis writing, but to check your understanding, I'm going to ask you to write a null hypothesis. This could be based on your most recent experiment you did in high school. It could be just a completely random one. But try writing out a null hypothesis for some research. For example, I might write that Year 9 students will show no difference on test scores, whether chewing gum during the test or not. It doesn't necessarily have to be a super rigorous hypothesis. If you don't want to operationalize your variables, I'm not going to have a go for that or anything. Want to make sure you understand the concept. If you do want to operationalize your variables, that's fine. Just saying I'm not going to be super clear. Right, exactly. If one plant's watered with tap water, another one with mineral water, there will be no difference in the nourishment of these two plants. So, you might be measuring that in something like growth or the color, but ultimately you're just we're not going to see anything going on here. We're going to get the same result. That's exactly what we want a null hypothesis to focus on. I did see some typing earlier, but it seems to have gone away, so I will just continue. Then we have two different kind of ways we can go wrong here, right? We can reject the null hypothesis despite it being true. So this is where we go, there is a relationship even though there's not. So, for example, if you're taking a rapid antigen test to see if you have COVID, a type 1 error would be where it says you've got COVID, but you actually don't. Whereas a type 2 error would be is if we accept the null hypothesis despite it being false. So for example, rapid antigen tests not detecting COVID even though you have it. With a rapid antigen test, they are much more likely to have a type 2 error than a type 1 error. So, while they're not perfect, very skewed asymmetry on there, where if the rat says you've got COVID, you probably do. If it says you don't, well, you might anyway, because the type 2 error is much more common. You can see that in this table here, where we reject the null hypothesis, but the null hypothesis was true on this type 1 error, and you can see this alpha symbol, which we'll talk a little bit more about. Of course, if we reject the null hypothesis and it's not actually true, great, that's what we want to do. And if we accept the null hypothesis and the null hypothesis is true, then great, we were right. So just want to make sure that in particular, we don't want to reject the null hypothesis if it's the case. We don't want to go thinking that there are all these relationships out there, this causes that when that doesn't happen. So we tend to focus a lot more on minimizing the type 1 error than on the type 2. And this is where p-values come in. So a p-value, if I'm going to talk about this really properly, it describes the probability under the null hypothesis that the test statistic is equal to or exceeds the observed value in the direction of the alternative hypothesis. So let's say we've got some data point here, some data point there, and then we're like, oh, what's the difference between these means? Then the p-value would go, okay, well, what's the probability of having at least that much difference in order to say that there's a relationship? Or if we wanted to investigate the possibility of having like something be broken, and we go, oh, okay, well, there were like, this many more broken ones under this condition, we would go, okay, well, what's the chance of finding that many more broken ones, even if this doesn't increase the chance of it breaking? So the p-value is saying, if the null hypothesis is true, what's the probability of getting this result or a more extreme one? And often the test statistic for this is the mean, or we're looking at the difference of two means. Often people will describe the p-value as being the chance of obtaining your results due to random luck. This isn't a fully accurate description, but it kind of gets the point across in terms of what it's conveying. And where p-values are often used and what people will often think about them in terms of is that if our p-value is less than alpha, then the results are considered statistically significant. Now, alpha is often set to 0.05. So you can have some differences there where, for example, we're taking a one-tail versus a two-tail statistical test where are you going, what if it's just more extreme in one direction versus testing both, where that can, alpha can be changed to reflect that. There's some other things we have to be wary of, but really what we're interested in is that we want to make sure that there's less than a 5% chance of obtaining those results under the null hypothesis. So if our p-value comes back and it's less than 0.05, we know there's a less than 5% chance that we would have gotten those results if the null hypothesis were true. So then we can feel confident in rejecting the null hypothesis and accepting the alternative hypothesis, saying there is a relationship between these two things. That's what our data shows, that's what we see here. This one should be a pretty quick poll. Researchers, generally, do they want their data to have a high or a low p-value? Remembering that if our p-value is below alpha, then we get to say our results are significant, reject the null hypothesis, accept the alternative hypothesis. Right, we've got our first answer in. Yep, we're in agreement here. Absolutely. Being for an agreeing, we have a low p-value is generally what researchers want. then you can say my results are statistically significant. If you put out your paper and you're like, oh, actually, we're not sure, maybe this was just due to chance, it could have happened if there was no relationship anyway, that's a lot less exciting. You're probably going to get less citations and have less impact. You want to have a low p-value, so you can say, look, this is meaningful, we do have the relationship, it's not just due to luck in how we were sampling, for example. So congrats everyone there. So if we're going to be specific with our terminology here, assuming our standard case we've got alpha set to 0.05, if our p-value is less than alpha, we reject the null hypothesis, accepting the alternative hypothesis, so we're like, yes, there is a relationship here. So, of course, it's possible we could be wrong, right? Maybe the null hypothesis was actually true, but we're like, it's unlikely enough that we feel confident saying this is statistically significant. So we now have what's called a significant result, and you don't want to use the word significant unless you're talking about this. You're talking about that statistical mean. If our p-value is not less than alpha, so we've got some bigger number, then we fail to reject the null hypothesis. And that language there is a bit careful, because we're not necessarily saying that the null hypothesis is true. We're saying, well, we can't be confident enough to say it's not. So maybe it is true, maybe it isn't, but we don't have enough confidence to say that there is a relationship. We're basically just like maybe. So depending on your results here, you might go, well, maybe with a larger sample size and then having more statistical power, maybe we would be able to reject the null hypothesis and say there is a relationship, but at the moment we don't have enough evidence either way. Often what people use and report are these 95% confidence intervals about their sample statistics. So they might have calculated their sample mean, and then they'll go, well, I can have 95% chance that the true mean, the population mean, lies within this range. So figuring out the size of this confidence interval is going to depend on what kind of statistical distribution your data follows, what sample size you had. Generally, the higher the sample size, that's going to give you more confidence about your statistics. And also the spread of values, right? You might have collected a lot of data on it, but this is something that shows a lot of natural variability, and so you've got a wide range anyway. Or it might be that, this is something that actually only tends to have a more narrow range. You could be pretty confident that the true mean is that, except that you didn't collect enough data. So when we're looking at this 95% confidence interval, we can link this back to the standard distribution, right? With the standard distribution, if our data follows that, it's normally distributed, then we expect that the value will be within 2 standard deviations of the mean. Usually we're not going to know the population standard deviation, so we rely on the sample standard deviation. So when I'm talking about the population standard deviation, I'm talking about the true one, right? If we could measure everything and have all of the data in the world, this is what we would get when we calculate the standard deviation. But we don't have all of the data, we only have our sample, and so that sample standard deviation is a different value. In this, we can use the standard error to quantify our precision around the mean by taking our sample standard deviation and then dividing it by the square root of our sample size. This reflects, the sample standard deviation, the variability in the data. If we didn't divide it by the square root of our sample size, that would kind of imply the more data we get, we're more likely to get some deviations from there. So we correct for that by dividing by the square root of our sample size. The more samples we've taken, the more confident we can be that we have a more representative sample and that we actually can see the true values. So again, really important here with language, if 2 groups have a similar mean, then there's a small effect size, right? So you might do your research on your tap water versus your mineral water, and then you find that the plants achieve similar height, similar color, they've got similar results from that. And so you'd say there's a small effect size, but let's say you did this to 1 million plants, of this particular type that you were investigating, then you could be pretty confident that even though there was only a small difference, if that difference is showing up again and again, then you're like, well, it's not due to chance. So you might have a statistically significant difference So it would be significant, but it actually is only very small. On the other hand, you might have, when you do this experiment and you find that the mineral water plant grows a lot higher, it's got a much more lush green colour that we can see, and we're like, wow, there really is a big effect from this, but you only tested out like 4 different plants. Your result's not going to be statistically significant, so the result's insignificant, even though there is a big difference. So make sure you're quite careful when you're discussing effect size and when you're discussing significance that you don't conflate the terms. So if you feel like you want to use the term significant but you're not talking about statistical significance, try going for something like substantial, meaningful, impactful, in order to talk about, whether the difference is such that it would really matter. Again, you can't use the term significant unless you're talking about statistical significance. Make sure you report your p-values. This is going to be included in your results. So if you have a figure with particular p-values attached to it, you would put that into your figure caption. Report your sample size. So this will be in the form like n equals, where you then have your number of your sample. When you read scientific papers, pay attention to how they report values and then kind of copy what the conventions are within your field. And I've said this before, but be careful with the word significant. When it comes to presenting your data in your results, table captions go above tables. So rather than just having like a table heading, what you do is you put a caption in that describes what your table is about, the key results from your table. You would put in brackets your p-value that you calculated. And when you show a table for like a scientific paper, you'll notice that they don't have those vertical lines or column dividers in there that sort of like, Word might automatically put in, or that kind of thing. Get rid of them. Right? You might get someone who's not fussy about it, but you might get someone who is, particularly as you go on past first year. Remove the vertical lines. Make sure, as always, that you carefully label your units. You want to put the independent variable on your left that has your different factors. For figures, figure captions go below figures. So for a table, you have to put the description above. For a figure caption, you have to put it below. Remove grid lines from the background. If in doubt, go for grayscale. So I had a bit of this shock when I was in second year bio, and we'd like handed in our assessments, and we were told, after we'd handed it in, why these in colours are meant to be in grayscale, and say I had no idea. So I'm telling you now, if you have someone who's sticking to like formal conventions, if in doubt, go for grayscale colours. Make sure you use clear axis labels and label units. Again, because you have the figure caption, you don't really need a like title over your chart. The figure caption takes place of that, but it's more descriptive. In terms of general best practice for figures, use a baseline of 0 to avoid being deceptive. Avoid pie charts. They're just a bad idea. Don't do it. If you have 2 quantitative variables, right, so your IV and your TV are both quantitative, you want to use a line graph. In order to differentiate it, if you have multiple lines going across there, instead of just using color, try using dashed lines, dotted line versus solid line to differentiate the different categories. If you're doing that, you will need to also include a legend to show what each of those are. And generally, this isn't a hard and fast rule, because sometimes your data might best be displayed using another scale, but use a linear scale. So your like y-axis values go up like 1, 2, 3, 4, or like 10, 20, 30, 40, instead of being like 1, 100, a million, a billion. Using a linear scale can make the data easier to intuitively understand. So I've got an example. This is some mock-up that I just made very quickly in Excel after dumping some data in there. It initially actually put them both as columns, which confused me because I don't, normally use Excel these days. What needs to change here in order to make this? So there's a fair few different things that are wrong, and I would want to change if I was putting this in a report. You can submit multiple answers, so you can just, chuck in one thing that's relevant and then go for another thing later. You don't have to do it all. Yeah, changing to grayscale is one thing I could do. It would make it more formal. In this case, because I'm not using the color for actual meaning, I would probably get away with it. And like it, really wouldn't be a big deal. But if I had something that was particularly harsh, then changing it to grayscale would be absolutely safer. And that way you're guaranteed to not be knocked for that. Scale so it increases linearly. So this one is actually a linear scale because it increases by the same amount each time. So see here how it's like 5, 10, 15, the increments are always 5. If it was a non-linear scale, it might go something like instead of 5, 10, 15, 5, 10, 200, 3000. Right, axis titles are required, so unit of measurement are known, absolutely. That's a pretty blatant one. What would I need to make sure I included in them? A couple of hints here. What's in here? Yeah, so with the temperature title, that's fine. There's something missing there. So you could probably imply, but like, do you think this is in Kelvin, for example? From the figure, we have no idea. We would want to make sure that we put the units in there in brackets. For this enzyme activity, would want that to go across to an axis title. We really don't need a legend here because we only have one thing that we're graphing, so that's redundant and would get rid of it. Basically, you want to get rid of anything that doesn't need to be there, because then that's just distracting from the interpretation of your data. So we would, get rid of the some activity label, put it on the axis title where it belongs. Temperature, and that's for our axis title, we need to put our units in there. Same thing, we'd want units for enzyme activity. We had them. What about this? Is this the kind of way to present a university figure? There's also something wrong here. I'm going to flick back because these are more university-specific changes. About these first 2 points. Do we have a figure caption on this? Should we be using a title or should we be using a figure caption? There should be a figure caption. Absolutely. So with this, temperature going on the y-axis, usually temperature does belong on the y-axis. Often it does. In this case, because we're looking at enzyme activity, which depends on temperature, temperature being on the x-axis is actually fine because temperature is acting as our dependent variable. But you are right that often temperature would be on the y-axis because often temperature would be our dependent variable. We would get rid of this enzyme activity title, would put it in a detailed figure caption. The figure caption kind of like, one way of thinking about it is that it stops people from taking your graph really out of context, right? You know, when people will crop things on the internet to try and make it like support their narrative. With the figure caption, that provides a bit of context to your figure, which then makes it a lot harder for someone to do that. That's not the only reason to have it there, but it is something that it achieves. Another thing is that I would get rid of these horizontal lines in the background. Again, this is something you might not be penalised for in, say, first year, but it could randomly sneak up on you at some point that you get marks taken off you for that. Generally, those lines in the background are a no-go because you don't need them and seen as distracting from the data. So if I wanted to make this properly, I would get rid of those background lines, get rid of the title, get rid of the legend, put in the axis titles with the units in brackets, and put down a more detailed figure caption. Alright, I'm going to close this off. Thank you everyone for your answers. As you can see, there are some things that you have to change that in high school that you don't have to worry about. And then we've got this question. With presenting data on graphs, what did I mean about having the IV on the left? Also, on a figure, you do want the IV on that y-axis, which will be on the left. But what I was talking about there is more tables. So if you're presenting a table of data, then whatever your kind of important factor is that you think might be causing these changes, your independent variable, you put in the leftmost column. Like don't put it in the middle of your table somewhere, put it over there on the left. Let me know or resubmit if that doesn't answer your question fully. So on this next page I've put in, this is a figure I made a couple years back. So you can see it's a bit of a different type of figure with a bit of information on it. I've got my labels here, I've got latitude and longitude, looking at a particular area. I've got my legend indicating what my different colours mean. Because I'm showing something spatial, I have a scale. And then in my figure caption, I'm saying, well, this is what it's showing. Right, so these are my eDNA proportions over the study area as predicted by the hydrodynamic model I was using. Colours indicating the eDNA proportion deciles. So that means that, one 10% grouping of the data is in red, one is in the yellow, one is in the green. And then I've indicated what the NA is referring to, and that the numbers at the top of each figure indicate hours past start time. So you can see the figure there, its caption is a fair bit longer than what a title would be, provides a bit more information. In this case I have used colours, but these are also colours that if it was converted to grayscale, you could still see the difference between them. So colours aren't an absolute no-no, but if you, it's best to not rely on them where you don't have to try and use some other form, and particularly if you're doing kind of Excel type graphing, grayscale is safer. Right, and now we are on to our general guidance about university section. So if you had any questions about figures, tables, any of that, please, do ask. And we're going on to, this more kind of standard general guidance advice. One of the questions that I can often get is about the difficulty of uni. So in my experience, generally if you did quite well in your high school studies, you might find that first year university covers a lot of the same topics that you've already done, and that you're relearning things that you kind of already know, particularly if you're focusing on subjects that you covered in high school. So for my first year, there was lots of stuff that I just already knew from high school. Then as the years go on, you specialise more into your field as you're introduced to new concepts. University also moves a lot faster than high school. So there was some stuff when I was looking at the like quantum chemistry topic, for example, that when I was doing that in VCE physics, we went for like weeks over that, but then when we did it in university, it was like kind of 1 or 2 lessons. Then you were expected to know it. So if you don't get it from the lecture, it's then your responsibility to chase that up and to study a lot outside of the lecture, use office hours, drop-ins, use the university websites to like ask questions. No one's going to kind of make sure that you're going slow enough that everyone keeps along. If a bunch of people are getting it incorrect, they may go back over it more, but they've got a tight schedule and, you need to make sure that you keep up. Make sure you have strong foundations. Missing content makes it harder to understand future weeks, semesters, years. So even if you are finding that a lot of the stuff that you're covering at a particular point you kind of already know, still pay attention, still attend your lectures, because they'll drop in some new things, and then if you don't have those new things, later on you're going to be lost. You don't really have scaling in the same way at university, so there are some subjects that most people will find easier, some subjects that most people will find a lot harder, and so if you're trying to look up information on these, easy subjects are often called WAM boosters, so that's WAM standing for weighted average mark. So this might be something like, that you've got an 80% average or a 60% average, or whatever it might be. You've also got another measure of grades, which is the GPA scale, which is where if you, kind of get a high distinction, that correlates to a particular number. If you get a distinction, that's a different number. The different cutoffs used for the GPA vary between universities. So if you're interested in something like postgraduate medicine, for example, they will assign different weightings to different things depending on the university because they use different cutoffs. You also may find that you get lower marks than you're used to in high school, particularly percentage-wise. It can be very easy to miss things and then get marks taken off and not know that you were kind of doing the wrong thing. And you're going to be surrounded by other university students, right? So a lot of people who might be less academically inclined may not have gone to university, right? So you're comparing yourself to a different sample, trying not to get caught up in like imposter syndrome or that kind of thing, and not being scared of what if you're like surrounded by oh no, everyone else is finding this easy. You don't know, they might be, they might not be, but it can be easy to have that kind of bubble. When it comes to your assessments, make sure you know how much each assignment is worth and prioritize. Sometimes they might make something worth 10% that is like not that much effort, or something might be worth 5% and it's really hard. So particularly if you're really struggling with your time management, know how much the assignments are worth, so you can think about how much time do I really want to sink into this one. Repetitive tasks like quizzes can be a source of free marks, as long as you actually do them. So particularly early on, you can have some quizzes that you can like redo until you get it right, or that the answers are like right there for you. So if you actually keep up with like weekly quizzes, that can be a very easy way of boosting your marks. If you miss it, each individual quiz might not contribute much, but overall they do add up. Labs are often marked and compulsory. Make sure you know when your labs are in your timetable. You do not want to miss labs ever. You would want to have a very, very, very good reason that you could then put in, because you can get marks docked not being there, or you're not there. Okay, now you don't have the data to do the assignment. Good luck with that. Right? So, if you do have to miss it, like chase the staff up and like talk to them about, see what you can do. But really plan around not being able to miss your labs. Lectures may or may not be compulsory, right? Like I've skipped lectures before, right? People tend to do it. But if you fall behind in lectures, you might get lost in labs and tutorials. So generally I would say if you're going to not show up to a lecture, maybe it's like at a really inconvenient time, or something like that, make sure that before you do it that there is a recording available and that you catch up on the recording before your next thing for that unit comes around. Like the next lecture, the next lab, the next tute, because otherwise you just might not understand what's going on. And make sure with any assessment that you pay very close attention to rubrics and criteria. Often, particularly at the start, it can be hard to interpret. So in first year they are a bit more late and tend to like build up the difficulty more. So initially your lab reports Might be less writing a full lab report and more, fill in the blanks. But make sure for any assessment you do pay a lot of attention to that. In university, depending on your university and your subjects, you can get a lot of group assignments. Now, particularly in early years, you're likely to be assigned to groups and not be able to choose who you're working with. As you've probably experienced in high school, sometimes you're going to have people who are not particularly motivated or tend to leave things to the last minute. May or may not be you, but you'll have a range of motivation in your group. What I really recommend is that you set mini deadlines and meetings up along the way before the assignment is due. So if you've got some big thing due in a few weeks, you don't want to just let that go by. As soon as you know who's in your group, figure out what socials you're going to talk to each other on, figure out where you're going to do your assignment, how you're going to divvy up stuff, and then once you've divvied up the roles, have check-in points. Right, so you have a meeting. In this case, I've used, how about we aim to have the dot points for our slides ready for our meeting next Monday at 2 p.m. Because you'll get people who do it at like 1:59 p.m. on that Monday, and who only really do it just before that meeting because you have that meeting in place. So you want to have these check-ins along the way. Also, so then you can kind of feel the pulse and go, all right, well, if this person's not with what they're contributing, I think that we actually might need to edit that, or they might need a bit more support in this. You want to be able to pick up those things early. And remember that you don't want to finish the assignment when it's due. You want to finish it beforehand so everyone has a chance to look over it and then provide any feedback, make any adjustments, and then submit it afterwards. Right? So you want to leave some buffer time at the end rather than doing it at the last minute. You may also sometimes find that if you're like doing an online upload, that when it gets close to the time and a lot of people are using it, you could have an issue, right? You could have some tech problem, and you can't always get a sympathetic response. Then it might be like, well, why did you leave it to the last minute then? In any group assignment, try to have very clear division of responsibility, so you know who's responsible for what, that you can all see each other's work as you go. Often group assignments are presentations, and they may require equal speaking time or response to questions. So don't assume that you can kind of have one person in the group then like say everything or answer all the questions. You may get marked as well on the spread and everyone contributing. Often there is anonymous peer assessment included, so this is that kind of thing that might be penalising someone if they're not pulling their weight, where you can knock them on the peer assessment rubric. Alternatively, it's where you can go like, oh yes, this person was so great, they put in full effort, all good. That will actually often contribute to marks. So you want to remain on good terms with the people in your group. Now on to lab reports. So keeping clear notes in labs can help a lot later. You'll find that you have different types of labs. Some labs are like wet labs, which is more like your kind of chemistry lab where you're messing around with solutions and that kind of thing. Sometimes when you have a lab week, particularly for less lab intensive subjects, it might be like, okay, well, you're kind of in that space, but you're doing a lot of stuff that isn't really super lab work. Make sure, sometimes the teaching assistants or TAs can be really obvious about giving, some clear hints about you want to make sure you do this or make sure you mention that. Write it down because you probably will forget unless you write it down. Again, being careful of conventions like table captions going above the table and figure captions going below the figure. Use your lab manual to help you. So often in week 1 you'll be given different resources like the lab manual, and you want to use that as guidance. When you're thinking about some experimental methodology, try to think of targeted improvements other than just increased sample size. So you can get a lot of people from high school focusing a lot on bailing like, oh, the sample size is too small. Try to think of other things. And in the real world, you're not going to have a sample size of 1,000 a lot of the time. So I think high school students can often be a bit extra critical of sample size. Try to make sure that you're really not just going on that, and you can think of other improvements as well. Lab reports tend to become more difficult, and you've got their kind of guide rails taken a bit away as you progress through your degree. With scientific writing, you may notice some differences between how you're meant to write in uni versus high school. Sometimes these are school specific, so you might find that a psychology student versus a physics student are expected to do things in different ways. There tends to be a bit more emphasis on written communication skills, so things like being succinct, having an engaging writing style where it's clear what you mean, compared to what there was in high school. If you're really in doubt, either ask or go for the more kind of clear and formal approach. You don't want to be formal just for the sake of using old language, but you don't want to be very casual. You can absolutely be penalised for typos, spelling, grammar, style errors, that kind of thing. Generally, people find it easier to pick up on their mistakes if they print out the document and read it that way. Some other tricks you can use are using Control-A to select all your writing and then changing it into a different font. Kind of makes you think a little bit differently when you're reading over your own work. Or things like changing the colour background just to try and make it look a bit more fresh to your eyes, because it's hard to pick up on errors that you've made. And for specific types of assignments in different styles, usually the library website will have a lot of resources where you can look at that and it will give you more guidance. If you have something that's going to be hindering your ability to complete assignments, this might be something ongoing or something acute. If you've got something ongoing, generally at the start, your degree, there'll be talk about some service available. So this can be under different names, but it could be, for example, disability support services. And so, if, for example, you have ADHD, this is something where you could potentially get provision for that and then have those circumstances considered. So depending on what particular thing, you might have different allowance made. If you think you might be eligible, check out the program and consider applying early. You would need to supply documentation, that kind of evidence. Absolutely do not leave any requests until after the assignment is already due if you can possibly help it. Say for example you have something serious happen with your family, which is affecting your ability to work on the assignment. Talk to the person marking it, talk to the staff about it as soon as you can, because the earlier you bring it up, the more likely you are to be able to get some form of provision for it. In terms of communication, again, this is a pretty big theme with university, where the onus is on you to reach out to staff, etc. if you need help. And try to do this early. And if you're trying to ask for guidance on something, generally help requests are better received if you can demonstrate you've tried to find the answers yourself. So you've checked the lab manual, you've read through your unit guide, or you've read through the web page, you've checked to see if someone else has already posted up the question. And then in the first year, particularly, you have very large cohort sizes, so often you might be told, ask on particular web page or Blackboard, Moodle, whatever it might be, and then only email for if privacy is involved. Again, if you follow those instructions, you're more likely to be better received and to actually get help with it. And then this is something that varies a bit between different staff members, but although Australia can have a very kind of casual culture in terms of hierarchical distance, there are occasions where I would recommend using respectful / suitably formal language. So for example, if I first was reaching out to a lecturer, I would be more dear such and such, instead of hey oh mate, can you help me with this thing? Just that kind of thing can make it better received, which is obviously better for you if you can then are more likely to get a response that you want. So, it's not someone's gonna ground you or something if you ask for help in the wrong way, but just if you follow procedure more, you are more likely to get that kind of quick or detailed or answer that you're looking for. In terms of careers, this is something that a lot of people don't think particularly much about early on, but science is a generalist degree, not leading to any particular job. So I highly recommend attending career expos relevant to your majors throughout your degree. Start early to give you an idea of what opportunities are out there and if you want to make changes to your course plan based on that. I majored in ecology and conservation biology. I don't regret it, but it is a field where you're much less likely to get a job straight out of the gate, compared to if you were looking at something in more like IT space. So, you want to just make sure that you're aware and can make informed decisions. And then this can also give you an idea of oh, okay, this particular elective might be more useful for me, this one would be less useful for me. And networking can be really helpful as well for things like internships and those opportunities as well. With resources, there are often a lot of resources available to help you. If you look them up, ask about them and engage with them. So again, this is something where sometimes you need to take more of that initiative than what you would in high school. C's get degrees is often said. With science, I would be quite cautious about that, because if you want to go further on into research, you will need to be doing well academically, and you need to have different benchmarks for different things. So I'm not saying you have to dedicate your whole life to study, make sure you get full HDs, absolutely not. But make sure you know what repercussions have, so you know what choices you want to prioritise. Again, things like exchange and study abroad programs may require careful planning, not necessarily always well advertised. If you want to go on exchange, if you want to do a study abroad program, try and look them up early on, so that you can make it easier to work with your course plan. Because you've got to do things like sorting out what units are suitable and that you could get credit for from your home institution. I recommend keeping some electives free in order to help you have more flexibility with that. And that's, just if you want to have those options available later on. Now I'm asking a question here that's not really seeming super related, but it will go on to something I say about resource. So when you're writing references, say you're writing a lab report, or you're writing kind of essay, what do you think the rules around references might be? Yeah, absolutely. Hopefully who needs references is an obvious false answer. You, a lot of the time, absolutely will need references. And yet you need to check the assessment criteria to ensure you're using the correct style. And this is going to vary between what different classes you're taking, where some things might require, like Harvard, I ended up using CSIRO style referencing a lot, but that's not common. Right? That's something that was more specific to the units I was taking. If you're taking psychology subjects, you might need to use APA a fair bit. Some things won't really care overly much, but you have to check the assessment criteria to make sure you're using the correct style. References should be free marks. You want to always be getting all the references marks, and be careful with that. One thing that would have saved me so much time, so many hours of my life earlier on, is if I used referencing software. So you can use referencing software that then you put your references in there, and then it automatically updates your references that you have at the end of the document. You just refresh it, and then it brings all of them in correctly formatted. You could then change your style, and it's going to automatically update them all, change your style, in-text citations, everything. So if you start to use referencing software early, it will save you so many hours of your life. You're looking at maybe a half-hour investment early on to learn how to use it properly, and then it will integrate well with Word, or you can have ones that integrate with Google Docs, and do your referencing for you pretty much automatically. And you just put in, okay, yep, I want to use this reference, I want to use this one here, and it does the work. So I really recommend that as an option. I didn't do it until honours, but it would have saved so much of my time. If you're obviously not sure about what that means or how to go about that, talk to the library staff and they'll be able to help you and point you in the right direction in terms of using that referencing software. Alright, I've put a bit of space here at the end for audience Q&A. I have gone through our content, so as until we hit 12:30, I will make sure I'm keeping on contributing things. whether I've got questions or not. Science can be very rewarding, also frustrating at times. Make sure you keep an eye on your goals to prioritize appropriately. For example, I think having a job during uni is important, often looks good, as well as being useful for a bunch of reasons. So you don't want to just go necessarily full academics, but make sure that you're able to prioritize in accordance with those goals. All right, let's go over to our questions. So at the moment, I've got a question: what referencing software do you recommend? I recommend using one that's popular at your university. So your uni will have different licenses for different ones. I used the Mensley one that interfaces well with the Nature Journal and of that kind of thing. So I used that, but my main recommendation is use one that other people at your uni are using, because then that way if anything goes wrong, you have any questions, or you want to share references, it'll be easier. So one thing you can do with it is that sometimes people upload a library or share across references that they've found, and then you can import that. So that's going to be easier if you're using the same software. So go with the one that people at your university use. All right. Since I don't have a question at the moment, until that comes in, I'm going to talk a bit more about the social side of things, since this isn't something I've really covered thus far. Good places for making friends are labs. So I talked before about going on committees, but with labs, if you stay in the same kind of lab group, which often you might be forced to do anyway, then those are people you're going to be spending time talking to, problem solving with, and so that's going to make it a lot easier. Lectures generally were a bit harder to make friends in. It's more like you'll probably sit with any pre-existing friends in the lecture, but often people who are a bit, not super confident with socialization aren't going to talk to the people next to them in lecture theatre unless you already know them. So labs are good places to make friends. Then tutorials or workshops, that other kind of thing, where you can talk about questions together, ask someone else what they got, and talk about that kind of thing. If you can share your socials early on, often you'll have at least a few people or a lot of people for any particular subject, make a group chat for that. So if you get into that group chat by knowing someone who's in it and them inviting you or whatever, makes it easier. If no one's made one, you don't know of one, suggest to other people you're in a lab with about making a group chat, so then you can discuss things for it. You'll find that people then also bring up assignment questions and that kind of thing in there. Key ingredients for friendship are that you want to have interactions, and so that's going to help you with that. Otherwise, if you want to get more into the social scene and not just on the academic side, in O Week they'll have a bunch of clubs and societies visible. If you are just a part of a club or society as a passive member, you might not find that it does too much for your social circle. If you go to a ton of events, that will help more, but if you're just passively part of it, won't really achieve much. But if you start going to the events, or particularly for smaller ones, or if you join on the committee, suddenly if you join a committee now, you have something you're doing maybe every second week with the same group of people, so that repeated interaction and the fact that you've now got to work on organising events, contributing ideas, that will really expand your social network. So one of the hard things with university is that because a lot of people have various different timetables, you will find that you have different circles for different things, because people in one unit might not have your next class, so there is a bit less overlap. One thing I would also say on this is that it gets easier as you go forward in the years, as you get more specialised into your major, and now you're not looking at lecture theatres with hundreds of people, but a smaller group that it's easier to get to know and to talk with. Also, just in terms of O Week and going up to clubs and societies, you can get a bunch of free stuff. Generally, if you sign up for the kind of student association membership, it'll give you a discount that can often make a bunch of clubs and societies free, and then they'll be handing out stuff. So, just in terms of like free food gotten back throughout the year, it can be a very cost-effective decision. Things where it's like a club for maybe like a sport or something can be a bit different. You'll have different types of clubs suited to different things, like cultural ones, sporting ones, on just focused on the academics on your course. But look out for the ones that you think you might be interested in. If you sign up, there's no obligation that you have to actually like go to a bunch. Another thing I'll say on the social side is that in first year, a lot of people are in the same boat. Some people will know people from their high school who are also going. A lot of people won't. Like for me, I had maybe one other person from my high school. They weren't even in my course, but they were like in science more generally, and we like ran across each other on campus once. So a lot of people are going to be in that position of not knowing anyone, and people are generally going to be friendly. A lot of people don't want to make the first move, but honestly, I have been the person who goes and like turns to the person next to me in the lecture theatre and is like, "Oh, hi," you've kind of learned a set of like basic questions like, "Oh, what are you studying? What do you have next?" That kind of thing. What did you do in VCE in first year? Like after first year, people don't ask that one, and then that just kind of breaks the ice, and then you're talking to each other. And if you don't have the confidence for that, that's fine. Lean on your like labs. A lot easier to make friends in like a lab or like a math tute compared to a lecture. The other thing is that you have group work as well, right? So when group assignments eventually come up, that's a group of people that you're going to be interacting with repeatedly. You might not necessarily be like, like all of them a ton and you want to be great friends with them, but then that expands your social circle a bit. And again, it just, you start off with the basic things about like, oh, how did you like go with like X or Y, or what else do you have, or like what do you want to do? And then it can progress from there. The main thing is try and get like into and like build up things like group chat, so then when someone does have a question, they are asking it to people and then you can have a conversation. Also, a lot of people get scared of like not knowing the correct answer or feeling a bit intimidated in that sense. A lot of people are going to be in the same boat. Alright, if you have any other questions, I recommend you try and get them in quickly because we are just about at time. So again, I wish you all the best. Best of luck with your studies. Thank you for attending. If you do have more questions that come up, you can ask like on the Uni Notes website and get responses from people on there. Could be both, other new students like you, but also people who have graduated university or who have been in uni for a while. And again, remember, you're here to learn, right? So try not to get too caught up on all the stress and everything. At the end of the day, like I think it's very cool to just learn more about science and develop more in that sense. You might feel very intimidated in your first lab, but it gets better, it gets easier, and it can be really good. I've got great friends and everything from uni, so I think overall, very positive experience, and I hope that that's your experience as well. Alright, farewell everyone.